I am Dorothea Lee. I am the Proteomics Sample Prep Research Associate here. I am the little fairy that takes your tube and turns it into data. So we're going to go over your sample and you, how metaproteomics sample prep works here at ArcBio, some considerations, mostly pretty similar to proteomics, but with meta, everything's automatically more complicated. So we're going to talk about that a little bit. So overview, we'll get through this. And for those of you online, if you are not attending the hands-on sample prep portion of the workshop, I do have a few slides about lab safety that we just need to go over here. Feel free to leave or we'll pause for questions before that. All right, so some common sample types you might all be familiar with. Gut microbiome, skin, oral microbiome like Gustavo talked about. Your human body has a lot of microbes, whether you like to think about that or not. And there's a lot to be explored there. Some of the ones that I think are really interesting, wastewater and seawater, soil and sand, which if you didn't bring your own samples, we'll be playing with a little bit of that today. And aerobiome, our friends at the Reardon Lab here, and some other groups. So with these, right, you're getting samples from a variety of environments. You're getting all sorts of other junk in there besides hopefully your microbes, your fungi, anything with the proteins that you're interested in. You're going to get a lot of other junk. Some of which is in soil in particular, humic content is a big problem. So we usually handle that with some strong detergents like SDS. See that a little bit later. Other proteins, right? So we're doing protein prep. We want to isolate our proteins, but a lot of times there's proteins that we know there. It's not going to blow your mind if I tell you have hemoglobin. It's not really what we're looking for. So sort of the plant analogy of albumin or hemoglobin is Rubisco. It's a big problem in plant samples. And so these are sort of big molecules that you want to think about that are going to get pulled down with your same precipitation chemistries, but they're going to cause problems when you're worried about depth of coverage, when you're wanting to see, oh, maybe I got, 500 micrograms of protein and 90% of that is one protein I already know is there. We don't really care. Keratin. For those of you who submit your own samples, I like to harp on this one because you are full of keratin. The air is full of keratin. Your skin, your nails, your hair, I have a lot of hair, all of it is made of keratin, and it is another highly abundant protein that we do not like to see. So change your gloves, use alcohol, wipe things down, keep your bench space clean. Enough on that. Low yield is a challenge that I'm sure a lot of us are familiar with. There's different strategies you can do to alleviate that. And another one that people kind of forget about are native proteases. So things that normally are doing their job, right? They're breaking down proteins, they're breaking down anything in your sample. However, if you leave your culture sample in the fridge for a couple of weeks, then those native proteases are going to do their job and they're going to eat up all your sample. And finally, PEG, sort of the polymer equivalent of keratin, polyethylene glycol. It is everywhere. It is probably in you, it is in plastics, it is in all of your consumables, but it is really not our friend when it comes to data acquisition. So try to avoid PEG wherever possible, but that's just one that we see sometimes and it can really interfere with your samples. So we'll go through. So our standard workflow here at ArcBio is pretty much the same for metaproteomics or regular proteomics. You'll have your lysis and precipitation step, right? So you go from maybe your raw leaf or a little teaspoon of soil, anything like that. And we want to get only the proteins out of that. We want to get rid of all the other junk and we want to break apart those cells really well. Then once you've got your proteins, there's a couple of different strategies for quantification. You might be familiar with a BCA assay or a Bradford. They're colorimetric things. You can also use a nanodrop and do regular UV-Vis. Thermos has some cool ways of getting around that as well. But basically, you want to know how much protein did you actually successfully extract. Hopefully, it's a lot. From there, our standard workflow, we usually digest 150 micrograms of protein. So you need to do that reduction and alkylation step so that which we'll get into a little later, so that we can actually linearize those proteins and make sure that our enzymes can work and digest them. And then digestion, you want to turn your proteins into peptides, and usually we add in a C18 cleanup step there for desalting. We have a really awesome, really nice instrument, and we do not want to put junk on it. So clean your peptides. So first details here, right? This is just the reaction scheme for the reduction and alkylation. Basically, you're going to reduce those disulfide bonds so that the proteins aren't super bound up and tight so that your trypsin proteases can actually get in there. And then you're going to alkylate so that that reduction stays permanent, so that they are really unfolding. Here we use, so that was my next slide. Here we use trypsin and Lys-C as a combination just to really encourage, because trypsin doesn't like certain residues. So we actually recommend a mixture of trypsin and Lys-C protease. Just if you're having issues with your miscleavages, that can be a strategy. All right, so metaproteomics, like I said, is just proteomics, but even more complicated. And so one of the ways that we can try to alleviate this really, really rich sample is by separation. So chromatography, right, our mass spec is coupled to a UHPLC. That's great. That's super helpful. And you can use different column phases. We typically use a C18 reverse phase column because that's what peptides like. And just on the mass spec side, right, your ion source also matters. That's not something you need to worry about as much here because you're not running your own samples, but something to keep in mind. Your chromatography will help separate your peptides out. So maybe if you've got a phosphopeptide sample, you've got a lot of really hydrophilic stuff that's going to come out. You might want to change your chromatography gradient as opposed to if you've got something that's super hydrophobic and maybe you want to get rid of some contamination. Maybe you've got a PEG problem. These are like things you can play with to try and separate out what your composition of your sample is a little more. On the sample prep side, what you can also do is fractionation. And so there's a lot of different steps in the process that you can apply that to. Let's see, what are some important ones? So FAMES, my poster is out in the hallway that I recently did at the ASMS annual meeting, which is using this extra attachment. It goes between your LC and your mass spec. And it's essentially a form of ion mobility spectrometry, which can help you to sort of filter out ions that you don't want, depending on the voltage, right? So sometimes they're going to want to travel in the particular voltage that you set it at in that field, or they're not going to be interested in traveling, and then you sort of separate it out. What compensation voltage you use, is the sort of the same as what chemistry do you use. It's all up to what you're looking for, but it is another way to isolate or separate. SDS Page, gels are great. Gels are a classic method of protein fractionation at the protein level, of course. IMAC and other metal affinity columns, that's another option that's pretty popular for phosphopeptides, that sort of thing. We've used it for zinc proteins as well. So that will give you sort of the fractionation of, does this protein bind metal or does it not? What else do we have? Magnetic beads, Gustavo's passion project right now. There's a lot of really cool antibody beads. There's other just chemistries. There's metal beads. There's things that aren't super well defined as to what they fractionate and what they don't. But the point is that you get two fractions. That's sort of the trend along here is it's not always selecting for something in particular. It's just separating. So same thing with high pH, like I was talking about with your chromatography, you can choose, do you want the hydrophobic stuff? Do you want the hydrophilic stuff? This is done usually at the peptide level. We use a kit for that, and it gives you the option of up to 8 fractions. So you could have, 8 runs instead of just one with everything really clumped together, or running, a 270-minute gradient. You can do some quicker runs with some less complex samples. And then same thing, Amicon molecular weight cutoff filters are a nice way if you only are interested in peptides from a sample, if you're maybe only interested in peptides of a certain size, that sort of thing. So I just wanted to provide a list of all the things you can be thinking about if your samples are too rich, if maybe you're seeing a ton of Rubisco, right? Maybe you can just run it through a filter. There's a lot of different strategies out there. And then, yes, other chromatography details. I'm going to brush over this because that's not anything we're going to be talking about today in particular, but other column types, other types of mass spec as well. So for today, these are the reagents we're going to be using. I just wanted to have a list here and sort of the standards in our workflow. So I like this thermolysis buffer, comes in a kit that we use, and it's pretty good for a lot of different cell types, mammalian, bacterial. We found that it works pretty well. Something to consider when you're at your lysis stage, right, is do you want to do chemical or physical lysis? Both of them have their ups and downs. Chemical, if you're using a lot of SDS, you're going to have to wash that out. It's going to add more steps to your prep. Physical lysis, do you have a bead beater? Do you want to sit there and vortex it for an hour? You know, there's ups and downs, or if you have an ultrasonicator, that's another great option. So we like to do a combination of chemical and physical lysis. It might be a little overkill, but it usually works really well for metaproteomics in particular, where you've got a diverse background of maybe you have some gram-negative bacteria that are easy to get rid of. Maybe you have some gram-positive, they're not so easy to break that cell wall down. Another one talked about protease inhibitors, right? This Halt's protease inhibitor cocktail, any sort of protease inhibitor, will give you a little more wiggle room to do maybe a longer physical lysis step without your sample eating itself. So that's always nice. And like I mentioned, we like bead beating right now. It's quick, it's pretty hands-free, and it does a really good job. We've seen for gram-positive microbes, things with some fungi even, we see more of those in samples that we've subjected to a lot of bead beating as opposed to ones where we just sonicate it briefly or vortex it. So it depends on what you're looking for, right? If you don't care about gram-positive, if you don't care about fungi, then don't worry about it. And then your precipitation method, there's some debate. You might see TCA precipitation, you might see acetone, you might see TCA and acetone. That is somewhat a matter of preference. Again, it's sort of another selectivity in the process. We found that acetone is the most effective and gives you the most range, as opposed to TCA will get a few things that acetone won't, but acetone will get a lot more that TCA won't. So more of a matter of preference, but I wanted to share what our particular workflow is because we see a lot of different sample types and we try to generalize for what works pretty well. And then from there, you know, this will be our first try. And from there we can specify. So we're going to do the BCA assay because it's another kit. Another really good part of standardizing your workflow is if you have the ability to use kits, that's awesome. SOPs are also awesome. Same thing, the reduction calculation will be a kit. And this is more for you to reference back if you want to know what we're doing. I'll also provide the SOPs for you. So one note is that the acetone washing step and the C18 cleanup and nanodrop steps take a little while. So I'm going to be doing those. I will be your sample prep fairy behind the scenes again. But if you are interested in watching that, you're more than welcome to stick around. That'll just be an afternoon deal because there's 2 hours to sort of wait. And I didn't want to make you all sit around to put some acetone in a tube if you don't want to. But if you do want to stick around, please feel free and let me know. All right. So for everyone online, this is a little shorter than Gustavo's, but thank you for coming, and we're going to go over some, I will pause for questions real quick. If there's anything from anyone. Yes, say.
For the metaproteomics, what ratio do you recommend between sample the protein to the tubes?
So we typically do about 150 micrograms of protein, and we do, let's see, I gotta do the math on this. We do 50 microliters per 0.2. So we do 10 micrograms of trypsin. So I guess a 1 to 15 approximately ratio of trypsin to protein for digestion.